Okay, welcome everyone. Large guests in the audience, right? Large guests. That's a word. So today is a very special seminar. I mean, we have weekly icon seminar. Every talk was very interesting. This week is also very special because it's not just icon, it's also icon and mechanical engineering joint seminar series. And we'll go over the reason why it was actually structured as a joint series of joint seminar. And so just a brief introduction of Professor Mark Spong. I think a lot of people already know. That's why this talk become very popular, as you can see right now. Professor Spong is currently a professor, right, at the University of Texas at Dallas in the Systems and Engineering departments, and holds the Excellence in Education chair at the same university. Professor Spong was also a faculty member back in UIUC. I think that was the institution before you moved to Dallas, and also before served as a faculty member at the Lehigh and the Cornell University as well. There are so many awards that Professor Spong received, but just to name a few, including the 2020 Rufus Oldenberger Medal from the ASME community, and also the year 2018, the voted lecture prize from IEEE CSS. But also, I think Professor Spong served 9 years as the president of the CSS. Is that no, not 9 years.
1 year.
1 year as a president. And also 2016 Nyquist Lecture Prize from the ASME as well, and 2011 Pioneer in Robotics Award from the IEEE RAS Society. So meaning that it's very, very impactful research across from the control theory and also from the Robotics Society as well. There are many awards, but I'll just keep it a short list. Yes. And today, before we start this seminar, I wanted to introduce the co-host of this seminar, Professor Ben Yao from the Mechanical Engineering, to go over what is the Oldenberger medal. So please welcome Ben.
Thank you. Okay, the Oldenberger Distinguished Lecture Series, right, was established to honor of Professor Rufus Oldenberger, which is a home professor of mechanical and electrical engineering at Berlin, and also the director of American Centers. So I think the reason we want to have that distinguished lecture is just a very rumor, right? He's a pioneer in establishing the control program in ASME. So that's, I think, Purdue has the tradition of system measurement control group, which is other institutions don't have. And also because of his personal effort, he knows 10 languages. So because of that, he's also able to fund nowadays the AFAC, right, International Federation of Medical Control, along with 14 representatives from different countries. So Soviet Union now. So obviously, in US, the American Medical Control Council is the member for that organization. So he is the founder of that. So that tells you the pioneering type of work he's doing. And if you want to know more, I think we also have the YouTube videos. I think I'll talk later on. So without taking much time here. Okay. So because of those extraordinary contributions, so the ASME established this Rufus Oldenberger Award back in 1968, the year when we were born. To recognize in some way, it's the most prestigious award given a person in dynamic system control by the ASME organization. So Professor Spong is the recipient of the 2020 Oldenberger Award. So we at Purdue, we have just foundation established. We have this to have this lecture series. So we normally invite the awardee to come here to give the annual talk. Professor Spong was kind to accept our invitation back in 2021, but because of pandemic, and we couldn't hold in-person the lecture series, and then some also due to transition, whatever. So we haven't done any lectures since then. So today we are very fortunate to have the Spong here, right, to restart the restart of our distinguished lecture series. So, Fred, to you.
Thank you very much. Wow, I'm glad I wore a tie today. Thank you very much. It's a really great turnout. Thank you all for being here. Yeah, I spent 24 years in Champaign, Illinois, where I could just drive over and visit my friends and colleagues here. I haven't been back here for many, many years. So it's really great to be back and see, still have a few of the old timers left, and some are gone and some are still here, but it's a great pleasure to be here. So as you can see, the title there, it's a controlled Chetaev framework for optical tweezer-based unfolding of protein molecules. So that it's actually this is a work based on a colleague, Ali Reza Mohammadi at University of Michigan Dearborn. Melika Golgun is my a master's student at UT Dallas. This is primarily most of her thesis work. And I'm not going to assume that you know what controlled Chetaev functions are, so I'm going to try to make this self-contained. Why protein folding and unfolding? It's interesting that my work has been in robotics, but you know, if you're in control theory and dynamical systems theory, you have a lot of tools that can apply to many, many different areas. And one of the reasons we got in this, we said, well, here's a problem that Ali Reza wanted to work on, and I didn't know anything about it, but let's try control theory and see if we can get some results here. So that's kind of where this started, and I'll try to explain as we go along what we're doing here. Let me give you a little bit of information about our Department of Systems Engineering at UT Dallas. We started this department in 2011, so it's a very young department. We have a small faculty, but it is distinguished faculty with Steve Yurkovich, we brought from Ohio State as department head. Reza Mohammadi we brought from Australia. He's doing a lot of good work in mechatronics and nano manipulation, atomic force microscopy, and things like that. Professor Vidyasagar, you may know from his books. I brought him to UTD. He's now emeritus. And Stephanie Adams is our new dean, and her area is her faculty appointments in system engineering. And we have some very strong young faculty there. Some of the research interests that we're all interested in: control theory, mechatronics, quantum control, nano manipulation. You can just read the list here. We have a lot of activities going on, and we have faculty have established some of their research labs in energy storage, in network control security. Lars is my lab, my laboratory for autonomous systems and control and robotics when I moved from Illinois. I reestablished the lab. So we have a lot of good facilities for research. And we offer, we're just starting a bachelor's degree in systems engineering. We started with a master's in joint with our School of Management. And of course, since we don't have a PhD program, we all advise students from other departments. We have no barriers. But because of our growth here, we have open positions. Steve Yurkovich, after 12 years, has decided to step down, so we're searching for a department head. So if you have any interest, if you're a graduate student finishing your degree and looking for a faculty job, send me an email. If you know of people that might want to be a department head, let me know to let them know that we're looking. So that's a little bit of background about us. This project is a joint between UTD and UM Dearborn. And again, we're looking at control theory applied to problems like protein folding and unfolding. And I'll explain what all these are about. So, protein folding is a process by which the protein's amino acid chain changes from a random coil into a specific three-dimensional structure. And of course, it's that final structure that allows a protein to function, to serve various functions, and there's thousands and thousands of different proteins in our body. But it's a very complex process, and sometimes proteins misfold, and that can cause many bad consequences for health, things like Parkinson's disease, Alzheimer's disease, and so on. So understanding not only the protein configuration, but also the pathway. Think of dynamical systems, think of the trajectory as it's converging to the folded configuration, is important to understand how these cells work for normal and abnormal functions. So it turns out that the folded protein, think of it as it occupies a minimum energy state. So think of it as converging to an equilibrium in dynamical systems sense. And so all these tools, dynamical systems and control, Lyapunov stability, control Lyapunov functions, we have a lot of tools that we can pull out to use for simulation, prediction, and also in control. I'm going to try to show how we can use some control here. And our first results here were in simulation. We looked at as an optimization problem. Robotics people have been working in this area for some time, because one of the early models, you think of a polypeptide chain like a protein, you have atoms that are interconnected by links with rotational joints. Sounds like a open kinematic chain. And so people have used robotics motion planning tools to predict and to study the motion, okay, the folding proteins. So robotics people were excited when they saw that some of their tools in motion planning would apply to this. Now, you also think of the forces of interaction between the molecules. Think of the links now as being a little bit like a spring. And so there's going to be forces involved and causing all these molecules to converge, and they end up converging to a minimum energy state, okay? So we did simulations of that, but we're also able to use optimal control to not only to compute the trajectories, but to respect things like constraints on entropy, which other people have not done in the past. Because you have lots of constraints on energy, entropy. You'd like to not only compute the pathway, you want to respect some of these constraints. What I'm going to talk about today is the reverse problem, and that's protein unfolding. So the protein unfolding is also important to study. It's less studied, but there are several processes such as protein degradation by what's called ATP-dependent protease. I'm not a biologist, but what this really means is these are enzymes that break down proteins, and they unfold sequentially and can lead to a mechanism for your cell to recycle proteins. If you scramble an egg, what you're doing is you're unfolding the proteins, okay? Proteins subject to heat will, they say, denature, they will unfold, okay? Now, what we're going to do is we're going to look at what are called optical tweezers, and we're going to use these to sort of mechanically unfold, literally to pull on a chain of DNA and unfold it. Okay. If you want to interrupt with questions, that would be just fine with me. Don't hesitate. But let me just give you a big picture, because this is a thesis work of my student, and it's basically putting together some interesting ideas from control and biology. Basically, we have a model for our optical tweezer, and I'll explain what an optical tweezer is. We're using a model that was done by Bassam Bami and Andy Thiel and one of Bassam's students on controlling particles in an optical tweezer. There's something called a worm chain-like model. I mentioned a kinematic chain model for a protein. Worm-like chain is another model that actually says, well, these are not rigid chains. They actually have flexible links between the joints. And so we're going to use a little bit more sophisticated model, but that comes right from this paper. And then I won't expect all the students to know what I mean by a controlled Chetaev function. I'll explain that. And our control design comes from something that's called a universal controller. If you've had Professor Corliss's nonlinear control class, you may know what a what's called the Artstein-Sontag universal formula for control. And we'll try to apply those ideas here. So that's kind of the big picture. So optical tweezer is a really interesting technology, right? And again, it's all new to me as well. But basically, it uses focused laser beam to hold and move microscopic particles, biological models, in particular nanoparticles. And basically, since light has momentum, then the laser light can actually transfer momentum to some small particle and cause it to move. So essentially, you focus the laser into some position, and then the particle can be induced by interaction forces to move. And if you change the focus, you can actually move the focal point of that laser, and you get the particle to move around. So you can control the motion of the particle that way. So you can also say you can actually attach DNA molecules to what are called handles, other strands of other molecules, and you can attach them to, say, another tweezer, or attach it to a peg here, and then you attach the other end to this bead, and that bead can be controlled, position can be controlled by controlling the focus of the optical tweezer, and you can, if you get it to move, you can actually stretch the DNA molecule. That's kind of what we're looking at here. So unzipping DNA and unlocking these polypeptide chains is one of the applications people are interested in. Okay. And this sort of mechanical denaturation, which means basically unfolding, provides thermodynamic and kinetic information about these molecules, helps us understand the molecules. So the model from the Bamier-Thiele work is basically Newton's second law, right? We have, if I take X to be the position of this particle, then I have mx double dot subject to a number of forces. S of t is the restoring force from the optical tweezer, and we'll see that actually acts like a nonlinear spring. We have viscous drag. This is in some viscous medium. And we have disturbances, thermal noise, and other things. Okay. Now we're going to take XR to be the distance from this particle to the center of focus. And since we can, by changing the focus of the optical tweezer, we can move that focal point around. So we're going to call that the control input. Our control input is going to be the position of the focal point, and we're assuming we're able to control it. Well, how do we do that? Another interesting technology is, it's fascinating. You can do that by what's called an acoustic optical device. It turns out that sound affects light in this application, because you have the whole system in some viscous fluid, and the sound changes the refractive index of the fluid, and that can change the focal point of the laser, that causes the laser light to bend, right, or change. So we have this acoustic optical device, which lets us basically control the position of the focus, and that's our control input. Okay, the actual spring force, the force from the optical tweezers, is basically a cubic spring. X minus U is the position of the focal point, which we're using as a control. Viscous damping is just typical proportional to the velocity. And we have some saturation. This only works over a very small distance. We'll assume that we're within that distance, so we'll just forget about that for now. And so this is our model and this is a model that the Bamier and Thiele paper used. Cubic nonlinear spring, viscous damping, and external disturbances. Now, it turns out that in a viscous medium, the mass can be negligible. You can basically neglect the mass because the viscosity is more important. So we can actually if you set the mass equal to 0, divide through by, take this, you have beta x dot. You can divide through by beta d, you can reduce that to a first order equation. X dot being your state variable. Well, that helps a little bit. Now, I mentioned this worm-like chain model. That's a little bit more sophisticated. We wanted to incorporate the sort of the dynamics. It's not just controlling the bead in the optical tweezer, but now you have the dynamics of the DNA molecule. And worm-like chain model was proposed to do that in this paper by Bustamante and colleagues, and they basically fit this function to the measured data, and it fits pretty well. Okay. The freely jointed chain model, like the kinematic model, is the dotted. Okay, but the actual data that they measure is a solid line, and the model they fit with these squares here fits it pretty well. So it's got this quadratic term in the denominator and so on. But we call that the worm-like chain model, and it's not our model, but we're going to use it. Okay, so as I said, we neglect the mass. We say we reduce it to a first order equation, but it's non-affine. If you know control theory, it means it's non-linear in the control. Non-affine systems are harder to control. And it's got the worm chain-like model here. So this is the model we're going to use. Although, not quite. Okay, we're going to, the control theory we're going to apply is based on an affine model. So we're basically going to linearize this with respect to the control. To reduce it to also an affine control model. And what we want to do is to have the rate of change of entropy at least bounded. So we're going to introduce a constraint. We're going to unfold the molecule, but have bounds, try to respect bounds on the rate of change of entropy. Turns out that's proportional to this x and x dot squared. So we're going to constrain x dot by constraining the control input. Okay, we'll see how that works. Any questions? Well, this is just pretty simple here. All we're going to do is basically expand our control in our function is in terms of the control u and drop the higher order terms. So we get our final model, x dot is f of x plus g of xu, so 0. And f is just, again, the cubic spring with the worm chain force. And g is given by this term here. Is a function of x. And of course, you have places where the control effort g becomes 0 and the control becomes inactive or ineffective. So we're going to have to respect some constraints there as well by design of certain parameters. Okay, so that's our model. Okay, first order, which is nice, but nonlinear and with some constraints. Now, this is for the students. I don't know how familiar you are with this, but you know basically Lyapunov theory, right? You want to investigate stability of an equilibrium. If you have a Lyapunov, how many know what a Lyapunov function is? All the students. So it's a function. Think of energy. Think of a Lyapunov function comes from energy. If the rate of change of that function is less than 0, then the function is decreasing, which means the energy is decreasing. So you prove stability of an equilibrium by finding this function whose derivative is negative. Okay, and so you don't have to compute. You don't need things like eigenvalues. It works for nonlinear systems. Now you can turn that argument around. If the v dot, the rate of change of energy, is positive, then the equilibrium is unstable. What we want to do is we want to make the equilibrium unstable because we want to go from the folded state and move it away from the equilibrium to stretch it. Now, you can, the proof is very easy. If v dot is positive instead of negative, then the equilibrium is unstable. But that's a very conservative condition, right? To have, for an equilibrium to be asymptotically stable, all the trajectories have to converge to equilibrium. But to be unstable, you only need one to diverge, and it's unstable. All the rest can converge to it, okay? It's like in a linear system, right? You can have 100 stable eigenvalues, you have one unstable eigenvalue, the whole system's unstable. So that's a very conservative condition, this idea of Chetaev's theorem. Well, for example, a saddle point, right? You will never find a Lyapunov function whose derivative is positive to find a saddle point, because we have some stable trajectories and some unstable trajectories, but the equilibrium is unstable. So Chetaev's theorem says, look at this region here, in this cross line. If I find a function v, not a Lyapunov function, just a function v, that takes on positive values in this region, arbitrarily close to the equilibrium, v dot only has to be positive in that region, not everywhere. So it's a much less conservative. So Chetaev's theorem says, let v be a function, like a scalar function, that is bigger than 0 for points that at least are arbitrarily close to the equilibrium, but not necessarily in a circle everywhere around that equilibrium. Then if v dot is positive in this region, then the equilibrium is unstable. That's called Chetaev's theorem. And we want control Chetaev functions. Now there's something called a control Lyapunov function. Okay, now you have your Lyapunov function, whose derivative depends on the control. It helps you design a controller. So a control Chetaev function, same type of a function. V is positive in some region, in some neighborhood arbitrarily close to the origin. Now we have the supremum maximum value over the control, A plus BU, where A is, you take V dot, right? A is dV dx, and B is times F, and B is dV dx times G. That should be positive. It's kind of like the V dot positive, but for values, all the values of the control, or the maximum value of the control. Now, we call that a control Chetaev function, and it's analogous to control Lyapunov functions that have been used a lot these days in walking robots and lots of applications. But notice what happens. We want this thing to be positive, okay? So if B is 0, then A has to be positive all by itself. So we have a condition, if B is positive and absolute value, A has to be positive. Otherwise, it doesn't qualify as a control Chetaev function. Now, what we're going to do, we're just going to take v of x equal to x. Yeah.
So you're assuming that the control system is stable. Right?
I'm sorry.
The own control system next time you can have to get that stable, right?
Right. We want to destabilize it, right? So we take v of x equal to x. Lyapunov would say x squared, but this we can take it to x is positive close to the origin. And that holds in our model. Remember, we had a g of x. That holds for x in some region that keeps g0 from going to 0. And there's an extension in a paper by it actually comes from a paper by Eduardo Sontag in the next slide. But you can also have a control Chetaev function where you have bounds on the control input. And basically, again, if we want the control to be bounded in some region less than k absolute value, then we take, instead of the supremum over all u, we take it over u in this region, same kind of condition. But now in this case, we have if b is 0, a should be positive, and we also should have a bigger than minus kb if the region is defined by k. I don't want to go through a lot of mathematical details. We have a paper on that. For our optical tweezer pair v of x equal x, well, we have a constraint that gain should be bigger than this value. And we have to guarantee that our initial conditions and our parameters start out respecting these constraints. That's all that means. Okay, now this is really paper from the FMOP paper, but they took it from Zontag. This is called a universal controller. Remember, we had the A and B, dVdx F, dVdx G. If I take my control to be minus F, and I plug in my A and B times B, where F turns out to be this, what looks like a complicated formula, but it's not, it's computable. Okay, we have parameters A and P. It turns out that for any P and Q satisfying these conditions, choose F to be this formula. And or 0 if B is equal to 0. And then you can show some work involved. That's a continuous controller. And in fact, it ensures that the equilibrium is unstable in some ball of radius R around the origin. Okay. And I don't have the Zontag paper reference. Okay. Now, on top of that, the laser beam in the optical tweezer, well, it generates heat, right? So we also have some thermal noise called run noise, and with some spectral density and some zero mean typically. And we also want to say what happens if I have this thermal noise. And for now, just think of it as you're adding some kind of a Brownian motion to your dynamics, a random motion. So now it becomes a stochastic equation. This Euler-Maruyama integration scheme, all that means is it's a Euler integration, which you know what that is. And Maruyama extended it to simulate stochastic systems. So we're using his algorithm, and we have our parameters here, which are taken from the literature, parameters that people have identified for some of these problems. And my student simulates this with no thermal noise. Then this is in micrometers, right? It's very small. The forces are piconewtons, and the position is very small. So the optical trap, the position of the focus, the control moves it up to some value based on parameters, and the green line is the how much the molecule becomes stretched. And so we make this equilibrium unstable, and we're able to stretch it up to some value. Okay. So the control is successful in that with this rather complicated model with what we call a controlled Chetaev function designed to do the opposite of what you do in stability theory. You destabilize an equilibrium.
So, if I understand right, so because you're trying to prove that there exists a region sufficiently close to the equilibrium, where in that region you're going to get pushed away from the equilibrium, right?
No. It says that in that region, B dot is positive. That implies that equilibrium is unstable. Unstable, that's at least one trajectory blows up. You might have stable and unstable. Right.
So if you were to consider a ball around the equilibrium, doesn't mean that the ball, if you start there, there's just influence that has not been made. It just means that within the ball, there exists a sub-region where there's just a controlling that happens.
It means within that, yeah, it means in that ball, you have a control that makes the equilibrium unstable. Just think of it like that. And it doesn't mean all trajectories are going to go. You like to think of the saddle point. Some trajectories are going to go away. You might have a stable manifold in that region. Yeah.
So this simulation, the fact that it drove it away, was that just luck? I mean, like there are some trajectories where it could have not getting. Well, you don't have to remove it.
Yeah, you know what, it's a first order system, so it's in the first order case, they're all unstable. Yeah, we don't have another orthogonal state variable, right? We have a first order model.
Right, okay.
So yeah, that's a good question, but it's,
Right.
yeah.
So I really like the idea of making things unstable. But it means that you basically use the control U right there to escape, but it actually converts. So is it something like a bistable at the end of the day, constant U just passively stabilizing to some, which is at 0.1? Is that?
Yeah, so, yeah, it doesn't go to infinity.
Yeah, exactly.
Right.
Yeah.
The equilibrium is unstable, but we're enforcing bounds on the control. It's a bounded control.
I see.
And so we have to have bounds on the control, which means it actually, I want to say that it creates a stable equilibrium up here,
Yeah,
right?
exactly.
Yeah.
So it has some bistability.
Yeah,
And
right.
it has a switching
I think,
property, so-
yeah, I think that's probably, that's gotta be what's going on.
Yeah.
Yeah. Okay, now the next simulation just adds the random noise, the thermal noise. And again, think of it as a Brownian motion, and the control jiggles a little bit. And the position of the molecule vibrates under noise, but it still stretches eventually.
Just going back to the last slide, do you think just a consonant will achieve the same thing?
I don't know.
But like you said, it looks like the implement the constant.
So I think, and this is one thing I've asked my students, what, can you compare this to just linear, local linear, a feedback linearization? There's a lot of different controllers. And she hasn't yet compared them to other controllers, but you think of, if I just take constant control to have a positive coefficient in the linear term, it's going to be locally unstable, I guess, right? But that doesn't respect any of our constraints on rate of change of entropy and these types of things. We still want to respect those, right? Right. So there's other controllers you could design, and control's not unique. But we do respect our given our specs to put bounds on the control.
To follow up with the control input here, when you increase it from 0 to the constant value, it actually descending by your system stays set by extra constant.
Right.
And not because constant input control will kind of more like just change the equilibrium. For example, if we put a big mass here, then bigger becomes right away become equilibrium. Everything will attract
Yeah.
here.
Right.
The constant control is like something like that.
Yeah.
The big mass here and
Yeah.
it
The
will
constant
affect
control wouldn't necessarily do this. It might destabilize the equilibrium. But yeah, who knows what it's going to do later. Yeah. All right, so thank you. I think we have plenty of time for more questions. So this is. Yes.
Is it possible that for some disturbances, the system, the equilibrium can become stable again? They want to destabilize it, but under some disturbances.
Yeah, I suppose it depends on the disturbance, right? Here we have a zero mean random disturbance, and that doesn't happen, but depending on the disturbance, you could possibly, yeah. I don't know what physically type of disturbance that would be, since I'm not a biologist. Yeah.
So I just want to confirm my understanding on the Chetaev function. So in one of the slides, when you're introducing the function, you just said the Chetaev function p of x equal to x itself. So I just want to make sure the reason why is because in the model that you have here, we already know that x is always non-negative. Because in general, for the controlling on our function, we always one of the requirements for p of x always to be like plus non-negative, and therefore we are going to like usually we are going to use something like x squared.
X squared, right?
Yeah. So in this case, I'm just wondering, in a general case, if it for the Chetaev function should that also be the square?
No, because the condition says that X should be positive arbitrarily close to the origin. Okay, you go on the other side of the origin, it's negative, but as long as it takes on positive values arbitrarily close to the origin, it qualifies as a Chetaev function. Martin?
Yeah, so for that simple example, basically the condition is to X dot positive for X sufficiently small.
That's right, it's a if you have X dot equal f of X, it's a 1D system. You just make F positive, and it's on,
Yeah.
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 So this is quite simple. I mean,
Yeah,
this-
you can find the two, you see that.
Yeah.
X dot positive.
Right.
Positive.
Right.
Yeah, is there any plans to actually do experiments?
We don't have optical tweezers available. We have some colleagues that may have some. I'd love to try to do some experiments along these lines. Yeah, that's we'd need somebody that experiments with DNA to join the team and tell us how to do it, but would be interesting.
It's amazing that you can actually manipulate. Molecules.
It is, yeah. We can manipulate protein chains and DNA strands, and I find it fascinating. I find the whole optical tweezer technology really quite cool, you know. And what we want to also do another next step would be to, you can't do it too fast, because to do it too fast means increasing the power, and you increase the temperature, and the temperature can destroy the DNA. So how do we regulate temperature and still get, kind of better performance? These are lots of open problems. Yeah.
The question about, you write out the system as a dynamic system. My question there, you also have several nonlinear term there, F0, F and G. Is it possible to come from a way to tune some weights into nonlinear terms, from system properties? Other than you can still could change it to this.
The model, to be tunable, you'd have to change parameters in the hardware, I think. This just comes from a model that people have used to fit the experimental data from the hardware. And so we don't have control to tune those parameters necessarily. But yeah, if you could do it, then they would have some effect on stability and transient
For
performance.
example, if we have photos, right? We could change the payload to change weight, maybe additional equilibrium.
Right.
Wonder whether, for the DNA stuff, whether we do the same thing, to change some system properties to change the equilibrium.
Yeah, and if you could make the stiffness coefficient negative, you could have an unstable that way, but we can't do that, I don't think. But yes.
In your system dynamics, you said you need XP. So do you assume the actuator can have like very quick response, because you at the end of the day you need to exercise something, some actuator to achieve XP.
So that's a good question. What we assume is that this is our actuator. Okay, it's basically based on another laser. It's modulating the refractive index of the medium that is surrounding the particle, and that has an immediate effect on the, you change your refractive index, the light bends, right? So the light bends, and that changes the focal point of the laser. So we haven't included, say, how fast we could do that, assuming that we could take that as our control input. I think it's a pretty good assumption.
So from another perspective, maybe it's like we trying to find some desired trajectory to track such that its desired trajectory is definitely dynamically feasible.
Yeah, that would be if you could actually specify, not only control the position, but control the trajectory, that would be very interesting. Yeah. Yeah.
Can you tell us a little bit about what experiments you want done and what has already been done, maybe with the different controls to do?
I'd have to take some time to find out everything that's been done. The Bustamante group that developed the worm chain lock model, I think they've done some work on, and others have done some work on stretching these molecules. And I don't know the kind of control they use on that, but what we'd like to do would be to have a device like this where we could, put a microprocessor here and implement our controller as a function of the measured position. You measure the position. You have a digital camera underneath this whole thing that measures the position. So that's your output, or you measure a state variable to try to control that position here using our algorithm. That would be basically, if you have a setup like this, it'd be fairly simple if we could implement an arbitrary algorithm into this actuator, then we could do the experiment.
So
I don't know if the off-the-shelf hardware, usually off-the-shelf hardware, it's hard to do that. You have to re-instrument it and
What
everything.
would you measure in the experiment?
You would measure the position of the bead to which the DNA is
And from that,
attached.
what would you extract?
Well, if the bead goes this way, the other end of the DNA is fixed. And if the bead's going this way, you're measuring the elongation of the chain of the DNA molecule. You're measuring the length of the DNA molecule.
I'm not sure if this is out of scope, but like in practice, when you do like unfolding of this peptide chain, do you just use one tweezer on one of the peptide, or is there potentially needed multiple ones that you apply at the same time?
You can do both. There are ways to attach it on one end to a fixed peg of some kind. You can also have two tweezers, and you can actually do both. You can do both in theory. Yes.
Yes.
Yeah.
So the idea is that the bead is connected to the folded pin using the laser focus to attract it. Right. So in a way that we found that some control that we can constant, meaning that it's kind of using its passive non-linearity of the spring to the skin. It's not basically because of just the control that I'm fighting with every particular stiffness.
Well, yeah, so the stiffness, I mean, just even in the linear case, stiffness is proportional to the displacement.
Exactly.
But one of the variables in the displacement is our control.
Yeah.
So we're actually changing the length of the spring. We get a larger force when we change the length of the spring.
Yes. I'm quite curious on that. The approach with bistable structure, because if we actually model passive worm as kinetic chains, we can have some kind of bistability structure there by putting a small input that actually makes it to both, so that it's not you actually pulling it, which is triggered.
Could be, yeah, we mentioned that earlier. Yeah, the question is, in these nonlinear systems, could you have some like a bifurcation phenomenon where you have, I don't know, at some point the whole equilibrium structure changes and it goes to a different equilibrium or whatever, right? That's all. It's an open question. As far as I'm interested. Yes, in the back.
You said when as a daily small can ignore it, and then the system becomes a first order situation. Like, because the time leading is small, then extra is leading behind.
I'm sorry, I didn't hear that.
Like, if your M is really small, then extra will be leading behind. Reason how, I don't know that. When you read that, nothing reducing super order, is that not changing?
I see, yeah.
How is that extra order?
Well, you're saying, divide through by m. If m is very small, then x double dot is very large,
Yeah,
right?
but then you.
But yeah, basically if m is 0, then it becomes we're looking at the m equals 0 case. Static situation. Yeah, then you're just balancing the forces here. Exactly right.
Online. So online said, thank you.
Okay. Yes,
I'm curious because I suppose the model is like, mostly have some uncertainty. Because I'm not sure how certain we are sure about the mechanism probability. Then is there any like your control method can have some uncertainty guaranteed, say, if this model dynamics not really accurate, but I can still unfold it forward.
the controller is based on this arts thing, on a, it's a formula, which I don't know how robust that is in general. But, yeah, the parameters are measured experimentally. And we got those from the existing literature, and we just, at this stage of the research was preliminary. We're just using those models and have not investigated any, we haven't done any sensitivity analysis or anything. The worm chain model was basically a curve fit model that someone else had come up with. And you're right, I mean, the robustness is always the first question that comes up. A lot of control work.
I think the reason we're most about it. All you have to do is assure them. Take some range from ground, right? That's what we did in this.
Right, so the expert on robot control. So robot control means if I can say with some confidence, I know the parameter, but I know the parameter is in this interval. And there are a lot of work, like Professor Corn and others that will give you a robust control. Yeah.
I have another question about the digitalization part. So can we view it as pumping energy into the system? So in that sense, will energy shaping, briefly talk about this, is like energy shaping be a
I certainly would like to say yes. You could try, because I like energy shaping ideas. Yeah, adding energy and increasing the energy to move it away from the equilibrium. I think that there should be some ways to do that. We in our control, in our other work, which was just controlling the bead, then we want stability. We were actually using a control optical function. We were actually able to keep the non-affine. We had a control that we didn't have to linearize. So lots of different things that you could try, and energy shaping certainly would be one. Yes.
When you're stretching the chain out, then what, are there any things regarding tangling and untangling?
I'm not a DNA expert. We're talking about a very small range of, distance. But yeah, it's a highly complex folded structure, and you're taking one end of it and you're stretching it. So how far you can do that, what happens to the rest of it? That's all, people are looking into that, right? I don't know how many people have, you're not gonna stretch the whole thing, what DNA strand is what, 6 feet if you stretch it all the way, right? So,
I think that's more like a question, how do you define, what do you mean by fold and what do you mean?
yeah, right, exactly. So, yeah. And there's a lot of local equilibria in these systems. So folding, it can go to different, depending where you start, you can go to different folding configurations. That's related to a lot of biological function of the protein. What we're doing is maybe you're just stretching a little bit, and from that you get some information about the properties of the molecule. Stretching it all the way out, that's probably not possible without cooking it. Good questions. It's
That's one last question.
Yeah.
So I think the cubic stiffness model, I think that's quite also interesting because people model the continuum test, the contact test, the Hertzian model, which is actually 40 point. Okay, so I think maybe this could be another insight. We're actually looking at the continuum. I think the last question is, what is the good advice for the people who actually love math and controls, for my students here? That's something that you suggest?
Control theory is somewhat unique. It's a branch of mathematics and a branch of engineering. So if you want mathematics, you can do whatever, you can study control theory and do all the math you want. I got a degree in math before I ever went into engineering school, so. But I think, in my case, because of my pure theory math background, I always like to see the application in the experiment. Just to see if the math really works. And it really works. The control theory really works if you do experiments.
So thank you for the wonderful comments.
Thanks to the computer.